the antisense technology for delayed ripening in tomato. Now, when tomato ripening, during the process of tomato ripening, there is overexpression of a gene called polygalacturonase. What is polygalacturonase, or what is polygalacturonic acid? It's the pectin constituent. Cell wall has pectin, and galacturonic acid, or constituent of the pectin. Many of such monomers are linked and gives firmness to the cell wall. Now, during ripening, there is overexpression of polygalacturonase that degrades the pectin linkage of polygalacturonic acid. Cell wall gets softened, leakage of nutrients. During ripening, tomato becomes soft. Now, what was that tradition there before that flavor saver or delayed ripening tomato? The farmers, all that supermarket chain, those who are involved in that, tomatoes are plucked from the vine when they are still green and firm, so that during the transportation, tomatoes are not crushed. So after transportation to the supermarket, there was addition or application of ripening hormone ethylene. So artificially induced ripening tomato. Now we get in the supermarket itself ripe tomato, and that is marketed. But there is some compromise. Although tomato is artificially ripened, there is reduced or low flavor, quality compromise. Low quality because flavors gets reduced. After that delayed ripening tomato, flavor saver tomato, that means tomato ripe but still it is firm. So there is no need of plucking tomato when it is green. It can be plucked after it is ripened, and then there is transportation. There was transportation, so there is no less loss during transportation because of crushing, and can be marketed. So there is increased flavor. The Calgene, they did what they did. They introduced, how they developed this delayed ripening tomato. They introduced the antisense, a gene containing antisense to that polygalacturonase. How we can do this antisense, introduction of antisense gene into the plant genome? Any idea? Cloning that gene in inverted repeat orientation, so that the opposite strand get transcribed. We have an RNA that is complementary to the original mRNA. So there is double-stranded RNA. That time it was thought that the double-stranded RNA prevents binding of ribosome. So there is translational inhibition. So that's why gene silencing. So in 1994 itself, Calgene got approval for commercial cultivation in USA. Then the big giant Monsanto acquires Calgene. That's a normal process normally in business, where their big company annex or acquire small company having some intellectual property. Monsanto acquired Calgene. Subsequently in 96 they stopped marketing of flavor saver tomato because it was less profitable. So that was the antisense technology that time. Then came during that time in 1990, when antisense was marketed in 1994, 4 years back, there was one Beezer incidence. The Jorgensen group, that paper actually Napoli et al. Published in Plant Physiology. Jorgensen group, what they were trying to do? They were trying to do deepening of flower color of petunia. Petunia has normal violet color, and in that anthocyanin pigment biosynthesis pathway, one gene is there, chalcone synthase. They thought of, if we overexpress that endogenous gene, there could be more intense purple color in the petals of the petunia flower. So they overexpressed. To their dismay, they found that they got a different shades of color in the petals of flowers. Some are less purple, some are variegated, and in extreme cases, what he got? Complete white flower color. They could not explain that Beecher incidence and termed co-suppression. Subsequently, in Fire and Mellow, they showed that it is due to RNAi. They coined the term RNAi because of double-stranded RNA-induced gene silencing. Double-stranded RNA is a potent inducer. This is very simplistic overview of RNAi. Double-stranded RNA is the potent inducer of RNAi. What happens to double-stranded RNA? There is an RNase-3 family endonuclease that cleaves double-stranded RNA into small interfering RNA, 21 to 25 nucleotide long, that is then incorporated into an RNA-induced silencing complex RISC that guides degradation of targeted mRNA by complementarity, based on complementarity. This is simplistic RNAi, gene silencing. That then RNAi was used for in post-transcriptional, transcriptional gene silencing. I am not going into details. This is the history. Now came genome editing. So what is the basic requirement for genome editing? We have to have a double-stranded break. If there is a double-stranded break in the genome, in a precise way, now with that in a precise way, in a perfect location, desired location, we create a double-stranded break. When there is a double-stranded break, what happens? There is repair mechanism inherently there in the cell. There are two processes of this repair mechanism. One is non-homologous end joining, illegitimate, not very precise. So it will take place. In that process, what happens? During that repair mechanism, either a few one, two, or a few nucleotides addition or deletion. So an indel is created. When there is addition or deletion of a few nucleotides, there will be frame shift mutation. Complete or different frame shift means a totally complete protein can be, so there will be knockout. Unless depends on the type of frame shift mutation that takes place during the repair process. In another case, when there is a template DNA homologous to that breaking site, so that template can be used as a template for the repair process, and there will be addition of a few nucleotides the way the template directs. So that is actually very precise genome editing. Addition in a precise way of few nucleotides. We will know again a precise way single nucleotide editing also. So this is the requirement of genome editing. Now the question comes, how many ways we can make double-stranded breaks? Any idea? We know CRISPR that makes. Apart from CRISPR, do you have any idea how many ways we can create double-stranded break? Yes. There is one more also. So we have different techniques of making double-stranded break. One is meganuclease, then zinc finger nuclease, TALEN, CRISPR-Cas. If you see the feasibility that increases from meganuclease to CRISPR-Cas, creating gene construct, handling, making double-stranded break. If we go from meganuclease to zinc finger nuclease, TALEN to CRISPR, the feasibility increases from left side to right hand side. Mine. So, we will discuss a little bit, touch upon each one of that. Before that, that is another way of doing genome editing without making a double stranded break. Let us discuss very briefly. That is called oligonucleotide directed mutagenesis. Heard about that? Here, the mutagenesis is incorporated in a precise or targeted way by introduction of small oligonucleotide, very short, maybe 20 to 50 or 100, very short, and that is without introduction of any other mutagen. Only it is per se oligonucleotide. That oligonucleotide is at the 5' and 3' end. There is a bit modified to protect from degradation. So, it could be single stranded DNA, double stranded DNA or DNA RNA hybrid. When that oligonucleotide is introduced either through bombardment, electroporation, what happens? That oligonucleotide having match to the DNA, it will make a displaced, the complementary strand, there will be local displacement, oligonucleotide will bind, and if we introduced one or two nucleotide mismatch for the repair to take place, the endogenous cell repair machinery will repair that mismatch and then introduce the desired nucleotide at that mismatch based using the oligonucleotide as template. And that has been used, introduced through this oligonucleotide mutagenesis, introduced in a number of crops. Basically, mostly for creating herbicide tolerance. And one such that is called RTDS, Rapid Trade Developmental System, that company Sivas, that is also commercially approved. Actually gene edited, probably first plant or crop that is improved in case of oilseed rape. You know, the acetolactate synthase enzyme is involved in amino acid synthesis biosynthesis pathway, branched chain amino acid biosynthesis pathway. Which one is the branched chain amino acid? Biochemistry friend, anyone? Isoleucine. Very important essential amino acid. That acetolactate synthase is involved in synthesis of branched chain amino acids. Now, they introduced a oligonucleotide that called GRON, G-R-O-N, gene repair oligonucleotide, that has homology to the particular site of that acetolactate synthase gene with one nucleotide mismatch. That changed one amino acid in the gene encoding acetolactate synthase, that makes it insensitive to sulfonylurea herbicide. So, through that GRON introduction oligonucleotide, that company developed oilseed rape that is tolerant to herbicide, sulfonylurea herbicide. The endogenous acetolactate was susceptible to sulfonylurea herbicide. But through a single amino acid change, that acetolactate synthase becomes tolerant to sulfonylurea, and they got. So, this is one example where there is no need of making a double stranded break. Only through introduction of maybe the efficiency may be a little bit less. But that oligonucleotide won't get integrated into the genome. Without any integration, without any introduction of any other mutagen, it is the function of oligonucleotide itself through induction of repair mechanism. Now with this, there is, have you heard about meganuclease or homing endonuclease? We heard about restriction endonuclease that recognizes 4 nucleotide bases, 6 nucleotide bases. But there are some endonuclease we call meganuclease or homing endonuclease that can recognize a very long sequence, more than 12, 15. In that case, I am referring one enzyme that is called ICRA-I. That was the first time used. It recognizes 22 nucleotide sequence. First time, actually the original ICRA-I that cleaves the 23S rRNA gene of chloroplast of Chlamydomonas. The algal, it called, belong to the Laglidag homing endonuclease family. It's a dimer that recognizes 22 nucleotides. Now, what would be the frequency of a nuclease that recognizes 22 nucleotide sequence? For 6 nucleotide, what would be the frequency? 4 to the power 6. Suppose in a genome of 10 to the power 9 nucleotides, what would be the frequency? 10 to the power 9 by 4 to the power 6. One just calculation. In case of our nuclease which recognizes 22 nucleotides, what would be the frequency? 10 to the power 9 by 4 to the power 22. Single or may not be. So it will recognize once in the whole genome. That is one such homing endonuclease belongs to that Laglidag endonuclease family. ICRA-I recognizes 22 nucleotides. Now if we, that's the wild type. Of course, then we need to have some variant to use as double stranded DNA breaking system. So then subsequently E. coli screened, E. coli library was made, later on Saccharomyces library made, its enzyme was crystallized with DNA bound to the DNA, and their DNA binding specificity were analyzed. Now then some mutant versions are created which can make a double stranded break. There are certain examples of using in plants also using this meganuclease as a system for making double stranded break. Bit difficult because we may not get such mutants which can recognize any sequence. Their screening, still screening is on both in E. coli library and Saccharomyces, so that we can have a different mutant versions which can recognize a different recognition sequence. And that should not affect its catalytic efficiency. So that is one which can make a precise double stranded break. Then came actually the first genome editing or double stranded break nuclease came zinc finger nuclease, ZNF. So zinc finger ZFN. We heard about zinc finger. What is that? DNA binding domain of many transcriptional factors. So zinc finger is a domain of a protein that binds to the DNA. It is around 30 amino acid domain, and it precisely recognizes 3 nucleotide sequence. Could be any. Now there are different zinc fingers were identified either through natural variants or through protein engineering that can recognize 3 different nucleotide combinations. How many combinations we can have of 3 nucleotides among 4 nucleotides that are present in the DNA? 4 to the power 3, 64. So 64 different combinations possible arrangement of 3 different nucleotides. So different zinc finger domains are there, available now through protein engineering and through natural variants. So if we can align different zinc finger domains, normally 4, so 12 precise nucleotide can be targeted of any sequence. Now zinc finger is for binding to a particular DNA sequence. We need some system for double-stranded break. Definitely we need to make two activities actually. One is precise binding to a DNA sequence and then making double-stranded break. So we need another system to make double-stranded break. They took help of a type 2S restriction enzyme FOC1. FOC1 is a type 2 restriction enzyme that recognizes 5 nucleotides GGATG and then make cut the upper strand 9 nucleotide downstream and the bottom strand 13 nucleotide downstream. So 4 nucleotide overhang. Got the point? Makes a double stranded break 4 nucleotide. The upper strand 9 nucleotide downstream, bottom strand 13. Make a staggered end cut, 5 nucleotides overhang. Now the FOC1 enzyme has 2 domains. One is the DNA binding or recognition domain, another is nuclease domain. So they took help of the nuclease domain and it is translationally fused with arrays of zinc finger domains. Got the point? 4 arrays of zinc finger domains for precise targeting of 12 nucleotide. That is translationally fused with nuclease domain of FOC1. But there is a catch. FOC1 nuclease domain becomes active when it dimerizes. So we need to bring another domain or another subunit of nuclease domain of FOC1. That is done by targeting the bottom strand or other strand, maybe 15 to 20 nucleotides apart, another zinc finger array if we can create through protein engineering that will bind 15 to 20 nucleotides apart downstream. When they will bind, it will bring the 2 FOC1 nuclease domain together and then induce a double-stranded break. And that way we are targeting a precise 12-12-24 nucleotide sequence. That will be very precise, sequence specific. So any sequence can be targeted. Only thing we need little bit protein engineering. Because we need to translationally fuse, first of all, 4 zinc finger domains that will recognize different 3 nucleotide sequences. If we have a 12 nucleotide sequence, so 3 into 4, so we need 4 different zinc finger domains to target that 12 sequence, and another 4 domains of zinc finger 15 to 20 nucleotide downstream that. So we need 2 constructs, in fact, for precise targeting of a particular sequence. That was actually first genome editing tools was reported first time. And using that, a number of genes being targeted. One is, phytate, the anti-nutritional factor. What it does? It chelates phosphate that was not inaccessible for nutrition, and there is, especially for the livestock, ruminants, it's a loss. It excreted out without absorption. Ultimately, that reach to the water body, there is eutrophication. So if we reduce the gene involved in phytate, it's a polyalcohol actually, phytate, that chelates phosphate. If we reduced, silence or edited a gene by any means of genome editing, so phytate will be less, there will be less chelating of phosphorus, and that will become accessible or available for absorption. So first time they did a gene called IPK1, that is inositol 1,3,4,5 kinase pentakis phosphate kinase 1. What they did, they targeted that zinc finger nuclease to make a double stranded break in the gene of IPK1. Then supplied a template DNA. That template DNA contains in fact one gene for herbicide tolerance. So that gene, whole gene got inserted within the coding frame or warp of IPK1. That IPK original gene got disrupted, but the herbicide tolerance whole gene was there, used that promoter of IPK gene, it got expressed. So two purposes were served. We got herbicide tolerant maize and less phytate containing using zinc finger nuclease. Another in case of that sulfonyl urea receptor gene of targeted, and they got resistance, tobacco resistant to sulfonyl urea herbicide by creating modified version of acetolactate synthase. Then came, we call now it TALEN. Many of you know what is tail effector. The rice causing bacteria Xanthomonas oryzae. During its infection, what it does, it introduces some effectors proteins, the avirulence protein, that goes and bind to the promoter sequence of some particular genes. We now call it sweet genes. Overexpresses sweet gene, original bacteria, pathogenic bacteria causes rice disease, Xanthomonas oryzae. That effector protein goes inside the cell, induces expression of some proteins we call sweet proteins involved in efflux of sucrose from the cell to the intercellular spaces, so that bacteria can use it for its survival. What they do, how they activate that gene, they go bound to the near that promoter region, recruits the transcriptional factors, proteins, induces the gene. Now we call that is the susceptibility gene of rice that helps that bacterial infection. Now what the tail effector protein? Now it has different domains for binding to the DNA of particular sequence. One domain around 30 to 35 amino acid long. And in that domain, two residues we call DVD, residue 12 and 13, that specifically encounters a particular nucleotide in the DNA. So each domain can recognize a particular nucleotide sequence. Now there are different arrays of these tail effectors, which at the 12th amino acid position has different amino acid 12 and 13 that recognizes a different amino nucleotide sequence. So if we put different tail domains, an array of maybe 18, so it will specifically recognize 18 nucleotide sequence, is not it? And then that is translationally fused to the nuclease domain of FOC1. So it will one hand again like zinc finger nuclease at two sides, 15 to 20 nucleotides apart, two sequences we need to target. So that will bind, bring the nuclease domain for dimerization and activation and make a double stranded break. Again, we need to do a little bit of more genetic engineering. go little bit fast, and a number of genes were targeted. The same gene which is responsible for helping bacterial infection, that sweet gene, sweet 14, that was targeted, so it's killing the pathogen with its own machinery. Use the talent to target that susceptibility gene which helps bacterial infection. That promoter sequence targeted, where the tail effector binds. Wheat powdery mildew resistant locust was targeted to talent to make it resistant to powdery mildew disease. And then soybean, in case of soybean, that fatty acid desaturate gene was targeted for its activation. Why? Now you know what is linoleic acid, what is linolenic acid, omega 6 fatty acid, omega 3 fatty acid. Which one is omega 6 fatty acid and which one is omega 3 fatty acid? Linoleic is omega 6, linolenic is omega 3. Both are 18 carbon. In case of linoleic, there are 2 cis double bond. In case of linolenic, 3 cis double bond. And this is linolenic is omega 3. Now why? Now it's very much essential for health, for prevent cardiovascular diseases, to keep heart fully functional. Polyunsaturated fatty acid we call PUFA. But why do we need to prevent that? Now for many of the industrial use, for baking, for making biscuit, they make hydrogenation of that polyunsaturated fatty acid. Now during high temperature frying, they were maybe breaking of double bonds, low fatty acid, low chain fatty acid, that's very dangerous, carcinogenic. So there is hydrogenation. In the process of hydrogenation, trans fat is produced. Butter is also one such hydrogenation product. Otherwise, polyunsaturated fatty acids are mostly liquid, normal temperature. Increasing hydrogenation becomes bit in normal temperature, hard. So makes trans fatty acid, that is very dangerous for health. So to make less polyunsaturated, which can be used for industrial purpose, they wanted to silence that gene involved in polyunsaturation. The fat gene is targeted through talent. And in case of potato, they targeted one gene, it's also very near to my heart where I am also working. That gene was targeted called vacuolar invertage. You know, potato, it's after harvest we keep in cold to make round the year, make potato round the year available to prevent its physiological vigor. Potato is harvested, it's in India it's a winter crop, short day crop. But if we can see normal the potato, it's actually long day crop. It was in South America, in Peru, where it is grown during summer month, long summer day crop. But subsequently after domestication we grow it in winter, harvested during February, January and February. Then there is long summer month, so we need to keep in cold storage. But during keeping cold store, what becomes? It becomes sweet. Why? That storage starch is degraded and converted to reducing sugar. In that pathway, amyloplast, potato starch degraded, transported to the cytosol as sugar, sucrose is formed, sucrose is transported to the vacuole, there it is degraded by the enzyme called vacuolar invertage and becomes sweet. Not preferred for eating. The most problem with sweetened potato is with a term called coal-induced sweetening. When we make it or use for industrial purpose, making chips, we love chips, smacking up potato chips. But if a potato is highly sweet, if you make it, fry it at high temperature, there will be browning. That called Maillard due to Maillard reaction, an non-enzymatic reaction where the amino group of free amino acid reacts with reducing sugars, and there is brown coloration. bitter in taste, not preferred by consumers. Another health effect is generation of potent carcinogen acrylamide. There is so much concern now. The more the sugar, the more the chance of synthesis of acrylamide. So, if that gene was silenced for amelioration of cold induced sweetening through talent. Now we come what you are doing, a very brief discussion because all you know CRISPR. CRISPR is the immune system in the most of the eubacteria, 50% of eubacteria and most of the archaebacteria against the invaders. Who are the invaders? Phages, viruses and some during non-compatible conjugation. And what are the other processes available for defense for the bacteria? During the phage infection, if we see during the adsorption process, bacteria tries to stop adsorption of phage. Then during the process of DNA injection, bacteria tries to protect itself. And we all know that during the restriction enzyme, methylation of its own DNA, so that there is degradation is prevented. And another immune system is through CRISPR-Cas system. Bacteria defends itself from the invading DNA by degrading the invading DNA. So, if we see the timeline, the CRISPR-Cas actually was known since 1987. Different components were subsequently identified, the pathway was characterized, and in fact, it was in 2012 first time two groups simultaneously reported exploiting the knowledge from CRISPR. They showed in vitro that any exogenous or targeted DNA can be targeted for degradation. You know who are the two groups? Jennifer Doudna group in the Caltech and Dr. Zhang group in the Broad Institute, MIT. And since then there are a long standing battle for IP protection of that between the two groups. So, you know already, there will be just brushing up, we have in the bacterial genome genes for CRISPR array, genes for endonuclease, that is different class of Cas3, Cas6, Cas9. So, there are three processes. One is acquisition of foreign DNA or called adaptation. Then there is biosynthesis of CRISPR RNA. Then we call interference, CRISPR interference. Now, during the adaptation or spacer acquisition, what happens? If we see the organization of CRISPR array, what is this genes? Now, first the gene for a leader sequence and one end. Then there is the repeat units, a number of repeat units. In between there is spacers. The spacers are actually DNA that were taken from the invading phage DNA or foreign DNA. And how that spacers are introduced in a very chronological fashion. So, there is a record actually what time that DNA acquisition took place. So, this is the very simplistic pathway of CRISPR. The CRISPR array gene, when there is DNA get introduced, the surveillance mechanism searches the foreign DNA. They search for some motif, what we called PAM. protopressor adjacent motif. When they get it, they cleave that foreign DNA, and a piece of foreign DNA where their protopressor or PAM sequence is there, that is selected for as spacer, or we call it protopressor that time. Then the protopressor get integrated into the CRISPR array. So it becomes in part of that bacterial gene. Then there is transcription of that CRISPR array genes. So CRISPR RNA biogenesis, processing of CRISPR RNA. So after processing of RNA, the CRISPR RNA gets loaded into a nuclease complex. And which nuclease complex, it depends on which type of CRISPR system there. So how many types of CRISPR we know? Six types, type 1 to type 6, and these types are again classified into two classes. The class 1, where the CRISPR system is mediated by multiproteins, type 1, type 3, type 4, they are belong to class 1, where multi-protein nucleases are involved in CRISPR interference. Type 2 or class 2, where single protein mediates CRISPR interference, that are type 2, type 5 and type 6. Now let us go to very quickly how CRISPR acquisition or spacer acquisition took place. We will mostly concentrate on type 1 and type 2, and our most interest actually in type 2 system, because that is used for CRISPR genome editing for crop improvement. What is the, in case of type 1, the enzyme that is involved, actually the cascade system. And there could be two possibilities. One is primed, misprimed rather, and another is NAHEP. In case of misprimed, if there is some match with the loaded CRISPR RNA, so that will bind, take the cascade complex along with Cas3, and then since from that point it will scan the entire DNA. So and then if they get PAM sequence, so the cascade will cleave, and then there is release of protopressor. Another is called NAHEP, that is mediated by, rather than cascade, it is actually the REC BCD system that makes actually cleaves non-specific single stranded break of the invading DNA, and that broken DNA now get incorporated into the cascade complex. In case of type 2 system, the Cas9, single nuclease Cas9 that scans the DNA, invading DNA for presence of PAM sequence. Wherever there is PAM sequence, it recruits the Cas1 and Cas2 for cleavage and release of protopressor. When there is release of protopressor, that Cas1, Cas2 now involved its acquisition or integration pathway. Now there is, crash array, there is leader sequence, and there is then fast repeat. Now all recently acquired spacers are integrated at the leader end. So there is chronological order. The protopressor, 3' hydroxyl of protopressor makes nucleophilic attack at the fast repeat, that is at the leader end, and single strand is displaced, and that process the fast repeat is duplicated, and within that duplication of fast repeat, protopressor gets integrated. So when there is protopressors, then there is transcription. In case of type 1, the single CRISPR RNA is generated. Both the repeat and spacer sequence get transcribed. So RNA contains repeat sequence, spacer 1, repeat, spacer 2, like that. So in that, the Cas6 make cut at the repeat sequence. So there is 8 nucleotides handle, and at the other end some single stranded RNA cleavage, and then processing. So there is spacer and 8 nucleotide repeat handle. In case of type 2, in the genome, in addition to the CRISPR RNA, there is another RNA, DNA or gene coding for another RNA called transacting RNA, that has complementarity with the repeat sequence. So there is bipartite RNA molecule generated in case of type 2. Got the point? 2 RNA molecule, there is complementarity at the repeat sequence. So there is, in case of type 2, this is here at the repeat sequence complementarity, so transacting RNA binds. So it's a bipartite RNA, where there a cleavage is done by which enzyme? Which enzyme makes double stranded break? RNase3. So RNase3 cleaves there at the repeat, where there is double stranded RNA, CRISPR RNA and transacting RNA, and some other nuclease cleaves at the spacer. So in case of type 2, the transacting RNA actually makes some structure that is recognized by Cas9. So that is loaded based on that recognition of that structure, uploaded on the Cas9. Spacer, based on the complementarity, targets to the invading DNA based on the complementarity. So spacer will bind to the invading DNA based on the complementarity, and then Cas9 will make it break, plant and break the double stranded RNA. Okay? If we see now the interference, in case of type 1, it's a multi-protein complex, and there is Cas3, which is actually makes the cut, and Cas3 makes a single stranded cut. But in case of type 2, the Cas9 makes double stranded break. Now if we see the different domains of Cas9, it's had alpha helical lobe that unwinds the DNA, and then there is nuclease lobe. In the nuclease lobe, there are 2 domains. One is RUBC or RUVC domain and HNH domain. And then there is a domain that binds to that CRISPR RNA, both CRISPR RNA and transacting RNA. And the 2 nuclease domain, HNH and RUV, makes specifically cut to that 2 strands. Now HNH domain cuts that strand which is anneal to the spacer sequence. The RUBC makes a break at the displaced DNA strand. Is it clear? So here, now we now from this knowledge that CRISPR RNA containing spacer and some complementary sequence with the transacting RNA. Transacting RNA gives a particular structure folding that is recognized by Cas9. Cas9, that spacer then guides Cas9 to go and bind a particular DNA sequence, and there is one strand is annealed with the spacer, another is displaced strand. HNH makes break at the annealed strand, and displaced strand is cleaved by RUBC domain. From this knowledge, how we can exploit this knowledge for using CRISPR-Cas system? Now this is the bacterial system. But how we can modify that system to exploit it in crop improvement? Now that was original CRISPR-Cas system, a bipartite DNA. Now it was found that, later on it was found that if we create a single RNA molecule containing spacer of 20 nucleotide at the 5' end, then there is that sequence for the trans-acting RNA having different loops, stems, that is recognized by Cas9, that can be used for making double-stranded break at desired site. So instead of 2 RNA, if we make a construct of gene that codes for single RNA, in that we need to change the spacer sequence of 20 nucleotide at the 5' end so that any DNA can be targeted. So we now have a variable spacer sequence that in frame we can attach the trans-acting RNA sequence. We can entice the Cas9 for making double-stranded break in the DNA. Now for the crop improvement, we now need what? 2 systems. One is gene for Cas9, another gene making for gRNA. We call guide RNA cassette. Now Cas9 is a very big protein. It is around 4200 nucleotide long. Original bacteria. We need to modify its codon for optimal expression in plant. Then we need to target that Cas9 to the nucleus for go and making double-stranded break. So we need a Cas9 gene with nuclear localization domain. Whatever Cas9 gene you use, have you checked its sequence? Those who are using CRISPR-Cas system, from where do you get? Those who are using Cas9, CRISPR, from where did you get that gene construct? Mostly from AD gene. Did you check? It has actually 2 nuclear localization signals, one at the N-terminal end, another at the C-terminal end, for targeting the Cas gene to the nucleus, because it has to go to the nucleus for making double-stranded break. So in one gene construct, we need Cas9 driven by normal polymerase II promoter, mostly 35S, and a terminator, NOS terminator, 35S terminator. So one gene construct. Another gene construct we need for gRNA. In that, the gRNA proteins gene sequence will contain spacer sequence 20 nucleotide and trans-acting RNA sequence. But that RNA need to reside in the nucleus. It should not come out after transcription. So we need a promoter. Which promoter? Polymerase III promoter. Normally, what does polymerase III? Transcription of tRNA, small rRNA, and some small nuclear RNA. In major cases, Pol3 transcript, some transcript retains in the nucleus. So we need a Pol3 promoter. In most cases, either U6 or U3 promoter are used for transcription of gRNA gene. And then Pol3 transcription, we need terminator sequence. Any idea how it does? Now when Pol3 polymerase III encounters poly U or poly A in the genome, 5 to 7, it stops transcription. So at the termination sequence we need poly U. So we need a Pol3 promoter, Pol3 terminator, and that gene. And in that only spacer sequence can vary, so that we can target. That is here. So what are the different types of genome editing we now have? Based on the technique we use, we have either SDN, side directed, we call SDN1, side directed nucleotides, where only either zinc finger or TALEN or CRISPR we made double stranded break or induced double stranded break at the targeted DNA. And then there is imprecise addition or deletion of a few nucleotides, indel. That is SDN1. And subsequent stages, if we do selfing, the transgene containing Cas9 and others we can eliminate, and only desired mutation we can keep. So we can, through breeding, we can make free of transgene. Another called SDN2, side-directed nuclease 2, where if we can introduce a small template DNA at the double-stranded break site, so very small template where one or two or few nucleotides addition in a very precise way. So that is called SDN2, where we introduce a template DNA coding for the whole gene along with its promoter terminator. So that is introduction of a whole foreign gene, new gene, that is SDN3. Now SDN1 and SDN2, if we can eliminate through breeding the transgene, the Indian government now consider it is without any regulatory systems hurdle. But SDN3 will be treated as normal transgenic. Now we have very less time, going very quickly. So this is now through that we used Cas9, there are certain concerns of having Cas9 of non-specific targeting. So that's why another gene that belongs to type 2, the type 6 actually Cas12, that CPF1 type 5 belongs to, that is used for, that has less non-specificity. And here the pressures is at the 5' end in case of Cas9, but in case of CPF, spacer is at the 3' end. Here it is 20 nucleotide spacer, there is more than 30. Cas9 makes a few nucleotides upstream of PRANG, double-stranded break. Here it makes a few nucleotides downstream of the PAM sequence. The PAM sequence in case of Cas9 is 3 nucleotide NGG, there it is 4 nucleotides TTN. And here it is a single RNA. So it's a makes double-stranded break in case of CPF1 with overhang. You have already today's class discussed about CRISPR-mediated base editing. Now, two terms we use. One is dead Cas or D-Cas, another is nickage. Have you any idea what are those? What is dead Cas? I just mentioned two domains, RUB, RUVC and HNH, two active domains. So in that each active domain there is a single amino acid change. At the N-terminal end that RUBC domain is there. At the 10th amino acid position there is a change from D to A, aspartic acid to alanine as the RUVC domain. And at the HNH domain at 840 amino acid position there is a change from histidine to alanine. And that makes the Cas inactive for making double-stranded break. So that is dead Cas, which can bind to the DNA but cannot make break. Another is called nickage, where in one domain only there is mutation, another is active. So it can make only one strand break. Using that modified Cas, if it is translationally fused with some deaminase, nucleotide deaminase, either cytosine deaminase or adenosine deaminase, so Cas will bind, won't make double-stranded break, but as it is fused with deaminase, that will either deaminate cytosine to, when cytosine is deaminated, it will be converted to uracil. When adenosine is deaminated, it will be converted to inosine. So, that will induce GC to AT transition. So, that is using CRISPR-Cas mediated base editing. Going a little bit fast, what is prime editing? Discussed today morning, where dead Cas is fused with some reverse transcriptase. Along with in the RNA, there is a long sequence that can act as template. So, here nickase is used, one stranded nick, and use PEG RNA. There, and then with Cas, it is fused with reverse transcriptase. So, when there is a double stranded break in the PEG RNA, it is a more sequence desired we want to incorporate, that is there, that at the break point which will act as 3' hydroxyl end, that will be extended by reverse transcriptase using that PEG RNA as template. So, we can introduce any desired sequence through prime editing. So, similarly, we can do CRISPR mediated RNA editing using not Cas9, but another type 6 Cas, Cas13, that targets or binds to RNA, not DNA. If that is translationally fused, and it is a dead Cas we will use, translationally fused with any RNA editing enzyme, there it is called adder, either targeting to cytosine or adenosine, that can be modified deaminated and modified nucleotide conversion editing. And through CRISPR, we can do either activation or repression of a particular gene. How? Now, if we use dead Cas, that will go and bind at the targeted position, and that is translationally fused with some transcriptional activator proteins. Heard about transcriptional activator, which recruits transcriptional factors and also RNA polymerase to bind to the promoter site. So, there is more expression of the gene. Now, if we use dead Cas, that is translationally fused with some activator protein. Initially, the herpes simplex virus activator called P60, actually it is 64, and that is called Viper system activator, that is fused, initially used, that was 64, but later on some more transcriptional factors called P65, RTA. In case of, let us go to this next one here, that initially the VP64 herpes simplex virus transcriptional activator was translationally fused to the dead Cas. So, it will go bind to the DNA promoter region, recruit transcriptional factors and RNA polymerase for activation of the transcription of the gene. Later on, some other transcriptional activators are used, that is called RTA, and in case of plant, that heat shock factor from the heat shock protein genes, that is also as a transcriptional activator that was used for activation of that gene. So, based on the spacers, the Cas will go and bind to the promoter region, and it is translationally fused with activators that will recruit transcriptional factors and RNA polymerase for activation of the gene. So, quickly, there is no need to say that it is CRISPR based on the feasibility of handling and using developing CRISPR-Cas gene constructs increasingly be used suppressing zinc finger or talin, and they are used, and there are number of genes are targeted. It is very difficult to catch which genes are being or getting updated to that developments. These are the only few example already I mentioned about, and there are number of shares for the different plants where CRISPR is being targeted. The largest share is rice, then tomato, potato, wheat, maize, other crops, and it's a huge market that is projected to have by 2026 24 billion market. Not only for it's altogether more huge rather than in case of plant, in case of animal system, in case of human therapeutics, diagnostics. So there it's a huge market, drug market. And then so far these are the crops that has been approved using CRISPR-Cas technique. Because we are exceeding time, so then ultimately it's a sweet end. Though pioneer, those who not discover CRISPR-Cas, but they show that CRISPR-Cas can be used for targeting any DNA in heterologous system or in vitro. So they got Nobel Prize. So I stop here and thank you for your hearing. If you have any query, anything, please let me know.
We can have queries within later sometime, so he is around. So there are some online participants. So I would like to ask if any of them have any question.
Please.
So if any of the online participant has any question, so please raise your virtual hand.
Hi sir, good afternoon. If you can hear me, myself Abhishek.
Just a minute. Yeah, please go ahead.
Yes sir, myself Abhishek. I have one particular questions like, sir, as sir was mentioning about debt
Can you
class
speak a
9.
little louder because your voice is very low?
Hello.
Or in the
Hello.
chat box, if you
Or
can
in
write.
the chat box you can write. Yeah,
Yes sir.
quickly.
Yes sir. Yes sir. I will write in chat box, sir.
What is your name?
Sir, my name is Abhishek.
Okay.
So if you can give your email along with the query, I can send you the
Yes sir.
answer, and anything you can put in the chat box along with your email ID.
Okay.
Or
Okay sir.
my
Okay sir. I will.
email ID, is it mentioned in the first slide?
We will mention.
So you can send me directly to my mail. I can give you answer.
Okay sir. Okay sir. I will do that, sir.
Any other specific question to Dr. Patnaik?
Sir, if you can hear me now. If you can hear me now.
What is that? How dangerous are unintended consequences of CRISPR editing? Yeah, there are some concerns because in the CRISPR, it's a 20 nucleotide sequence. Although we try to generate this spacer sequence such a way there is no match in the genome. When we design the spacer sequence or 20 nucleotide sequence for targeting a particular region of the DNA, we use that 29 sequence for blasting the whole genome so that there is no off-target sequence. First, that need to be checked, and as per the report, the Cas9 has some inherent non-specific cut that is not precise, or we cannot predict at the beginning. So when we develop any genome edited plant, we need to see is there any unintended effects through phenotypic evaluation, or if need whole transcript analysis we need to do to see. And because of that concern, now as I mentioned, that Cas12 CPF that has a long spacers more than 30, that is used, and that has more specificity. Okay. So any other query you can send me to mail, and thank you very much.
So on behalf of all the participants, I thank Professor Patnaik for delivering his talk. now on behalf of all the organizers, I would like to felicitate him for coming to our.
Thank you for this nice gesture. Thank you. Thank you. Thank you. If you have any query, anything, you can come to free time. You can come to me and discuss.